Hello, welcome to The Moment. This time I have a single story to talk about, but one that I think is very interesting because it relates to a new discovery in medical research. If you have a story that you'd like to see addressed, please comment below. And with that said, let's turn to this week's story. Scientists at Newcastle University have made a breakthrough that could be very important for the future of eye surgery. The team, headed by Dr. Ricardo Gouveia, a postdoctoral research assistant, found that they could grow artificial corneas in a way that produces a far clearer product than currently. This involves growing the cells over a dome-shaped substrate to end up with a curved cornea, better matching the natural shape of the cornea than the currently made flat artificial corneas. I spoke to Dr. Gouveia about the discovery, and I started by asking how the method works.
So what we found was that by using different geometries for the support of the substrate where the cells grow, we can make them spontaneously align. And when the cells align on a curved surface, then they also deposit quite aligned matrix, which is the main component of connective tissues such as skin or the cornea in this case. The importance of this alignment is that it is really, really fundamental for the optical properties of the corneal tissue. That is, it needs to be robust on one hand, but transparent on the other. And this, by simulating the alignment that we have in the natural cornea, we can obtain tissues in a dish that have similar mechanical and optical properties.
I then asked how the current method for growing corneas was different.
So what people have been doing traditionally is to just grow them on a tissue culture plastic surface that is fat, and the cells in those surfaces tend to respond differently. So they grow in random orientations, and because of their orientations, the material, the matrix they produce and deposit is also randomly oriented. And that makes the tissue more fragile, and that makes the tissue more opaque, less transparent. The other thing that we found was that by inducing cells to align, they also respond differently in terms of the quality of the matrix they produce. That is, the matrix is mainly composed of collagen, fibrillar collagen, long and very robust fibers of collagen that resemble ropes, if you will. But there are other components that are produced between the collagen molecules that are contributing to also the transparency and the mechanical properties of the tissue. So incredibly enough, the cells that are growing on a curved surface and aligned because of that, they also produce more of these very specific components of the cornea, which is definitely a considerable advance over the existing techniques that are implemented all over the world. Now, we are talking about the cornea in this case, but there are many other tissues that need to be produced in vitro as a substitute for different organs. And what we don't know, but what we would like to explore next, is the ability of other cells, not just the corneal cells, but other cells to respond similarly to this kind of geometry, surface geometry.
I then asked how long they had been working on the project.
So this has been, I would say, the culmination of 5 years of work. We started by investigating certain kind of sophisticated, smart materials to be used to coat tissue culture substrates, and these materials basically allow cells that grow and produce tissue in a dish to be easily recovered after the tissue is formed. And then it's naturally evolved into trying to simulate a tissue that more closely resembles the shape of the cornea. So the obvious insight is it's obvious, but apparently no one had thought about this before. But the obvious solution is to try to grow the cells in a surface that emulates the shape of the cornea. And quite remarkably, we saw that the cells and the tissues that are formed on such surface reproduce what we have in the natural organ. Now, this can also constitute an interesting perspective on how, during development, during our own embryonic development, the cornea forms. So we think that this strategy will also allow biologists that work in development to investigate the effect of curvature on the behavior of cells during the development stages of humans.
Oh wow! So it's got quite a lot of implications outside of this area of research, then.
Yeah, that is true. So, and that is something that I really enjoy very much. So, the fact that you are working on a very specific area, in this case tissue engineering, doesn't mean that your discoveries cannot have much more global implications, much more wide implications to other fields of research.
Now, the project was undertaken alongside the University of California, and I asked Dr. Gouveia what they had added to the research.
We know many researchers all around the world that are working on corneal research. It forms a small but quite well-knit community, let's say, and these researchers have a microscopy technique called second harmonic generation confocal microscopy that allows to investigate the degree in which collagen fibers are aligned in a tissue. So we collaborated with them to understand the degree in which our tissues had collagen alignment and what were the differences between this alignment in tissues produced in flat or curved surfaces. It is definitely something that helped the publication to achieve a more impactful outcome.
I then asked when corneal replacement would be necessary.
Well, you do have several issues. There are genetic diseases that do affect the corneal stability, the stability of the stroma, which is the inner part of the cornea that is mostly comprised of the collagen of collagen matrix. One of which is keratoconus. So that is still a disease whose causes are still quite mysterious, but it involves the thinning of the cornea, and then with that thinning you have deformation and loss of the cornea's shape, and then loss of vision, and eventually blindness. And then you have environmental factors. It can range from severe dry eye disease that can then affect the cornea and lead to loss of corneal stromal integrity. And you have then mechanical and chemical injuries. One cause that has been quite publicized in the media recently is chemical burns that have been affecting many people here in the UK. These burns affect mostly the outer layers of the cornea, but it's still very severe, because if you affect the outer layers, it can also create an opaque layer of cells that will basically make you blind. But more severe chemical burns can also affect the cornea stroma, so be more penetrating and affect the integrity of the corneal stroma. There are many cases of these kind of chemical burns in India, but also in the UK.
I went on to ask when Dr. Gouveia thought the artificial corneas might be ready for medical use.
We showed before that these tissues are safe and easy to integrate in animals, in rabbits, using a rabbit animal model. We think that it would be very interesting to try to translate this technology into a clinical product, but there is still quite a bit of research to do before this. One of which is to create a method to make thicker tissues, and this is probably going to be done through the stacking of several of these tissues. The other issue is, of course, we need to understand what is the long-term outcome of such a transplantation. So, using a rabbit model, we show that the smaller pieces of tissue can be quite well integrated in up to 9 months. But in order to make sure this is a safe transplant material to be used in humans, this kind of study needs to be extended in time and also in scope. So probably by doing full corneal transplants, using these tissues, and not just using small portions of it.
Finally, I asked what other projects he'd been involved in.
We also look into the role of the mechanical properties of these tissues and the influence that mechanical properties have in the behavior of epithelial cells. The epithelial cells, the corneal epithelial cells, are the cells that I spoke before, create a thin layer that is the outermost part of the cornea. And this layer is quite important because it keeps the eye hydrated and at the same time protects the cornea stroma of entry of pathogens and other invasive bodies. So what we are looking at is the influence of a different rigidity of these corneal substitutes on corneal epithelial cells. Because in this paper we also show that depending on the degree of alignment of the matrix, we have a more or less rigidity. And by playing with different rigidities of the tissue, we can instruct cells that are growing on top of these tissue equivalents to respond in a different way. So it's a quite finely tuned mechanism that we still don't know what is underlying, what is the molecular pathways underlying it, and we are involved in that study.
The paper written on the research and results was published in Advanced Biosystems on the 20th of October, and I'll leave a link to the full paper in the description if you want to read it. Section 2 of the paper explains the method they used to make the curved corneas. It says, quote, "In order to create cornea-shaped tissue equivalents, we developed a novel template design with a curved geometry. First, lens-shaped agarose gels were cast using glass molds and subsequently mounted in tissue culture plates using a thin extra layer of agarose. This method showed to have three advantages. It secured the cured agarose gels in place, it created a flat region surrounding the curved templates where cells could be initially seeded, and guaranteed a smooth slope transition between the flat and curved surfaces, thus easing cell migration upward onto the curved templates." So that's the explanation for how they induced the corneal stromal cells to form a curved shape, and as Dr. Gouveia said, that particular shape gives the corneas improved strength and transparency compared to those grown on a flat plane. And in fact they can prove this further when they go on to say, quote, "Intriguingly, cell migration on curved surfaces was uniformly centripetal from the periphery up toward the apex of the curved surface template. The cell migration was unexpectedly highly organized and assumed a radial alignment all around the curved surface template. This behavior starkly contrasted with the migration pattern over the planar templates, where cells moving to the center showed a more expected random orientation." And I think that particular point is very interesting, as it would appear to indicate that this new curved method better aligns with the cell's natural behaviors. So, depending on how the test phase goes, this new method could be implemented in the very near future, which is going to be a good thing not only for the potentially millions of people who benefit from it, but also for the scientists and the future of research in this field in general. And that's all for this episode. Be sure to join me in a fortnight's time for more from around Northumberland and Newcastle. Until then, I'm Oliver Ralph, and this is The Moment.